Hello everybody, let us now study antimicrobials and bacteriocins. Food antimicrobials are chemicals which are added to or are present in food that retard microbial growth or kill microorganisms. Functions of food antimicrobials are to inhibit or inactivate spoilage microorganisms or pathogens, and a number of compounds have been approved by international regulatory agencies as well as regulatory agencies of various countries as antimicrobial for use in food. The chemical, that is the natural chemical, or there is other form of chemical to be used in a food as an antimicrobial should have certain characteristics. And what are these characteristics? These are, that is, the chemical should be less toxic or it should be non-toxic. Okay. It should have its antimicrobial activities over broad spectrum ranges, particularly the bactericidal as well as bacteriostatic effect of the chemical should be well established. It should be stable to heat and storage. It should have acceptable taste and odor. It should not only act against vegetative cells, but it should also act against spores of trouble creating microorganisms in food. Because see, that is the bacteriostatic, bactericidal and bacteriolytic, these are the three effects, that is the effects of the chemical, that is they can result into the bacteriostatic, means you can see here in this figure, that is the bacteria comes under the stationary phase, whereas the bactericidal becomes that the viable count of the bacteria, that is bacteria is killed, that is it decreases, whereas the bacteriolytic means total cell count, that is the both, that is the viable count and other count, they are completely killed, they are lice that killed. So, their count decreases, and these of course very important, that these antimicrobials should be effective in lower concentrations, and the concentrations at which they are required to be added into the food to get the desired effect should not result into any undesirable taste, flavor and other things into the food material. So, these are some of the important characteristics which should be there in the chemicals in order to qualify it for being used as a food antimicrobial. There are, however, certain limitations in the use of food antimicrobials. For example, the pH of the food, effect of the pH. You know, in earlier classes we have discussed that organic acids function at low concentration only in high acid foods, like the food which has pH less than 4.5. So, the most effective antimicrobial form is the undissociated acid, and which exists in the majority of the fruit only at a low pH or at a pH below the pKa of the compound. And you can see in this table that pKa, that is dissociation constant of most of the acid which can be used as a antimicrobial agent, they have the pKa less than 5, that is acetic acid has a pKa of 4.75, propionic acid 4.87, sorbic acid has pKa of 4.75, So, they have the all approved organic acids used as antimicrobial, they have pKa less than 5, means these acids will have maximum antimicrobial activity in high acid foods only. The food products which have pH more than 5.5 or greater, there are very few compounds that are effective at low concentrations, okay, because in these foods even very high concentration of these acids might be required, and which ultimately may have influenced the sensory characteristics or taste of the product. Another limitation to the use of these food antimicrobials, that is their interactions among the food constituents, that is these antimicrobials, they get their effectiveness gets reduced because of their interaction with the food components. For example, most of the antimicrobials are amphiphilic. So, they can solubilize in or be bound by lipids or hydrophobic proteins, and because of this they are binding with this hydrophobic proteins or lipids, they become less available for their antimicrobial activities. So, these two factors, like they can be used only at a lower concentration, or they become effective at lower concentration only in high acid food, or their interactions between the food components, even it may reduce their effectiveness. So, these are the two major limitations for the use of these antimicrobials. The various antimicrobials, that is which are of natural sources, okay. These, for example, acetic acid, that is the vinegar which is prepared by fermentation of the cane sugar juice, that is naturally it is found. Benzoic acid is found in plums, prunes, cranberries, cinnamon, cloves and many other berries. Lactic acid is found in the is produced by the action of lactic acid bacteria in dairy products and in other many food materials. Propionic acid is formed in Swiss cheese by the Propionibacterium, or sorbic acid is found in raw berries, rowan berries and all those. So, there are very natural antimicrobial compounds which can be extracted or which are obtained from the natural sources. So, these antimicrobial food antimicrobials are many times classified depending on the basis of the source from which they are obtained, like the antimicrobials obtained from the animal sources includes chitosan, lactoferrin, lactoperoxidase system and lysozyme. Those obtained from the plant sources include LM, that is hydroxycyanoic acid, isothiocyanates or spices and essential oils. The antimicrobials of microorganism sources are the food natural antimicrobials produced by microorganisms, include natamycin, niacin, bacteriocin and cultured products. Let us briefly discuss their characteristics, major characteristics, like for example, lactoferrin. It is a primary iron binding compound in milk and colostrum. Lactoferrin has 2 iron binding sites per molecule, and because of this iron binding activity, it becomes inhibitory to a number of microorganisms, and therefore, it can be used to prevent the growth or even to kill or inactivate the microorganisms in several food products. Similarly, the lactoperoxidase system LPS, it is an enzyme that occurs in raw milk, colostrum, saliva and other such biological secretions. This LPS reacts with thiocyanates in the presence of hydrogen peroxides, and this forms the antimicrobial lactoperoxidase system. The fresh milk contains about 1 to 10 milligram of the thiocyanate per liter. Hydrogen peroxide in fact is not present in the milk. So, approximately 8 to 10 milligram hydrogen peroxide per liter of the milk might be required to be added, for the generation of the LPS or lactoperoxidase system. So, in LPS reaction, SCN is oxidized to the antimicrobial OSCN thiocyanate, which also exists in equilibrium with hypothiocyanous acid, and its dissociation constant is 5.3. So, with this equilibrium, it results into the inhibitory activity. This provides the inhibitory activity against several microorganisms in both gram positive, gram negative and bacteria and others. Lysozyme, the enzyme that catalyzes hydrolysis of beta 1,4 glycosidic bonds between the N-acetyl muramic acid and N-acetyl glycosamine of the bacterial cell wall. This lysozyme is present in egg, milk, tears and other secretions. It is also present in insects and fishes. In hypotonic solution of the enzyme, the lysis of the bacterial cell occurs. Lysozyme is used to prevent gas formation in cheese, that is we call generally blowing is the defect in some cheeses. So, there lysozyme can be used, and maximum, that is the permissible limit or maximum limit which is recommended for use is 400 milligram of acid per liter of egg, per liter of egg white. So, that can be used. As far as the antimicrobials from plant sources are concerned, there are large number of compounds have been identified, they have been isolated, and in fact, their effectiveness against the microorganism has been proved. Lot of reports in the literature are there, even in very traditional or ancient literature also, the use of spices and spice oil extracts as antimicrobial system or antibacterial system or for various medicinal purposes etc. has been reported, like allium in onion and garlic, hydrocyanamic acids and related compounds in caffeic acid, para-coumaric acid, ferulic acid and synapic acids etc. The glucosinolates and their derivatives like isothiocyanates, spices and essential oils like eugenol in cloves, cinnamic aldehyde in cinnamon, linalool in sweet basil, vanillin in vanilla, all these are reported to have antimicrobial or antibacterial activity, and they are used as a food antimicrobial. So, in this slide I have just given you a list of the food antimicrobials which have been approved by regulatory agencies in different countries, and even this list may be exhaustive, a few more might be there in some other countries, some of them they might not be permitted in some countries. So, but a general list is provided here, and these includes alkyl esters of para-hydroxybenzoic acids like parabens etc., acetic acid and acetate salts, benzoic acids and benzoate salts, dimethyl or diethyl dicarbonates, lactic acid and lactate salts, lysozymes, natamycin, niacin, nitrates and nitrites, phosphates, propionic acid and propionate salts, sorbic acid and sorbate salts, sulfur dioxide and sulfide derivatives, medium chain fatty acids and esters. Nitrites are generally used in heat processed meat, poultry and fish to control growth of toxin produced by Clostridium botulinum. They are also used in some cheeses to prevent gas production by Clostridium butyricum and Clostridium tyrobutyricum. They are inhibitory to Staphylococcus aureus, Escherichia, Pseudomonas, Enterobacter, etc. So, these nitrates, they react with the enzymes in vegetative cell as well as with the germinating spores, and they result into the restriction of bacterial use of iron and the interference with the membrane permeability, and this result causes the this limits the transport processes in the bacterial cell, and ultimately which results its a growth process, or it means that the nitrates when they are used, they again disturb the homeostasis of the cell. There are certain side effects of the nitrates are reported in the literature, that it can lead to formation of carcinogenic compound like nitrosamines. So, there is a trend to reduce the use of nitrites for the preservation. Sulfur dioxide and sulfides are used in soft drinks, fruit juices, lemon juice, beverages, wines, sauces, pickles, fresh shrimp, etc. These sulfur dioxide are produced by undissociated sulfurous acid that rapidly enters the cell wall, and reacts with thiol group in the structural proteins, enzymes and cofactors as well as other cellular components, and this disturbs the normal physiological activity of the cell. It acts as a fungicidal at low pH and low water activity. SO2 acts as bacteriostatic at low concentration and acts as a bactericidal at a comparatively higher concentration. Hydrogen peroxide has a strong oxidizing effect on the bacterial cell wall. It can oxidize sulfhydryl groups of cell proteins and membrane lipids. Hydrogen peroxide producing reactions scavenge oxygen, thereby creating anaerobic environment that is unfavorable for the growth of certain microorganisms. Antimicrobial activity of hydrogen peroxide is enhanced by the presence of lactoperoxidase and thiocyanates, as we have seen in the earlier slide, that is it contributes in the formation of lactoperoxidase system, and which has a good antimicrobial activity. Diacetyl is identified as the aroma and flavor component in butter. It is produced by species and strains of genera Lactobacillus, Leuconostoc, Pediococcus, Streptococcus, etc. It is more active against gram-negative bacteria, yeasts and molds. It reacts with the arginine binding protein of gram-negative bacteria and thereby interferes with the utilization of arginine by the bacterial cell wall, and which influences its growth. So, earlier we have discussed, let us again see, that is how these organic acids they act, how they perform their antimicrobial activity. Okay. That is the weak acids have more powerful antimicrobial activity at low pH than at neutral pH. Acetic acid is the strongest inhibitor and has a wide range of inhibitory activity, inhibiting yeast, mold and bacteria, and it is the undissociated molecule which is toxic form of a weak acid. So, the undissociated form of organic acid diffuses across the cell membrane. Acid dissociates in the cytoplasm and releases the proton, and ultimately leads to the acidification and dissipation of pH gradient over the membrane, causing the observed growth inhibition. So, this is the way how these antimicrobials or how the organic acids perform their antimicrobial action. And in this slide you can read later that is there are different antimicrobial like acetic acid, benzoic acid, natamycin, niacin, nitrates, propionic acid, sarvicates, sulfides, etc. and their mode of action, how they, and as you can see here majority of them work by disrupting the cell membrane function, whether it is bacteria, whether it is yeast or mold, majority of them they function by disrupting the cell membrane permeability or cell membrane functions or disturbing the cell homeostasis. Another very important aspect comes when we discussed about this natural food antimicrobials etc. is the biopreservation. That is biopreservation is the, you can say, methodology to extend storage life or shelf life and to enhance safety of food using natural microflora and are their antibacterial products. So, in the food either the microorganism in live forms or the bioactives which are produced by the microorganism in this cell, it can be extracted and purified, that can be used as a antimicrobial in this biopreservation methods, or even there is certain like in the dairy, in the milk, whey etc. There is the where the microorganisms can be allowed to grow, and this fermented, that is the liquor or concentrate containing the bioactives which are produced by the microorganism, they can be used. So, this it has a wide ranging application in food processing. So, there are certain antimicrobials of lactic acid bacteria which can be used or in some time many times which are being used for biopreservation, and these includes organic acids like lactic acid, propionic acid, acetic acid or other low molecular weight compounds produced by these lactic acid bacteria when they grow in the food etc. or deuterin, diacetyl, hydrogen peroxide, fatty acid, cyclic dipeptide and phenyl lactic acids. And another important, more important that is component is the bacteriocin, and these bacteriocins are actually antimicrobial peptide, and they kill or inhibit growth of closely related bacteria, and the bacteriocins are gram positive bacteria are seldom active against gram negative bacteria. So, for that is the selection of culture to be used as a biopreservative, certain points must be considered, certain characteristics must be seen in that culture, and what are those? That is, this should have the culture which is to be used in the food as a biopreservative, it should be able to produce antimicrobials in the food, it should not affect even that the antimicrobial that is produced by these cultures should not affect, or even culture as such should not affect the sensory characteristics of the food. It should result into the limited number of acid or other bioactive productions, okay, should have weak protease activity, that is it should not adversely affect the protein of the food or other components of the food, limited gas production and absence of slime production. So, the microorganism it should have these type of, it should not have that is the characteristic which can influence the food components and food. As I told you earlier slide, that is the functioning of the biopreservative, these microorganisms which are the protective cultures, they can be used in the food either in the final product or even in the pre stages during the processing, or even they can mix with even the raw materials, and they result into the inhibition of growth of the pathogenic or spoilage microorganisms, or they can result into the extension of shelf life based on targeted inhibition of the specific spoilage microorganisms. The protective cultures which are used in certain food operations or food process as biopreservatives, they should be grass, or they should be listed in the grass. There is no indication of a health risk of this bacterial group and which is used as a biopreservatives, right. And the use of lactic acid bacteria in biological preservation is recommended, and even these use of lactic acid bacteria like lactobacillus species, lactococcus species or propionibacteria etc., they might even contribute to the health benefits of the products, because they may even provide probiotic effects etc. into the food material. They result into the shelf life extension, as well as they also can provide probiotic effect. So, factors promoting the use of bacteriocins as a biopreservatives are listed here in this slide. They do not alter acceptance quality of the food and are safe for human consumption. They provide effective barrier under wide pH and temperature ranges. Their activity is not lost in the presence of food additives and effective in dairy food during storage. They are effective in low concentration. The advent of novel bacteriocins with the broad spectrum of activity from food grade lactic acid bacteria has also made it possible the use of these bacteriocins etc. antimicrobials into the food. Even the consumer resistance to traditional chemical preservatives and concerns over the safety of the existing food preservatives like sulfides and nitrates etc. has also led to the more use of these bacteriocins. Safe and efficacious use of niacin for more than 40 years in several countries is reported. So, these bacteriocins, another important factor, that they are economical and they are safe to use in the food products. Some of the bacteriocins produced by lactic acid bacteria are given in this slide, like lysine A and Z. Niacin A and Z is produced by Lactococcus lactis subspecies lactis. Pediosin PA1 ACH is produced by Pediococcus. Then leucocins is produced by Leuconostoc species, and so on. So, there are many lactic acid bacteria which can produce bacteriocins. So, that is means that is one way, that is these microorganisms they itself can be used in the food, and where they produce these various low molecular weight compounds, acids etc., and which provides the beneficial effect in extending its shelf life. Another is this, that is they can be grown in certain foods and they can be extracted, purified and then used. However, the only commercially produced bacteriocin which is used at present is niacin, which is produced by Lactococcus lactis subspecies lactis, and Pediosin PA1 which is produced by Pediococcus acidilacti. So, the use of purified bacteriocin, however, have to be labeled as additive and they require regulatory approval. For example, niacin is approved for use as an antimicrobial in food by the joint FAO-WHO expert committee on food additives. The bactericidal action of niacin, let us see, there is niacin inhibits synthesis of peptidoglycans and also it interacts between the lipid 1 and lipid 2, and this inhibition of the peptidoglycan synthesis as well as its interaction with lipid 1 and lipid 2 may result into the pore formation, and which finally results into the inhibition of cell wall synthesis. So, they can bind the lipid 2, which is the main transporter of the peptidoglycan subunits from the cytoplasm to the cell wall. It prevents correct cell wall synthesis leading to cell death, and they can use lipid 2 as a docking molecule to initiate a process of membrane insertion and pore formation that leads to rapid cell death. Antimicrobial packaging is another very common use nowadays, that is active packaging, where these bacteriocins are used. Incorporation of bacteriocins into packaging films is used to control food spoilage and pathogenic microorganisms. Antimicrobial packaging films prevent microbial growth on food surface by direct contact of the package with the surface of the food. However, in order to make this use more wide and commercially more viable, there are certain methods that is there. One should have that a system for evaluating the effectiveness of these antimicrobials. That is, one should determine the efficacy of the compound in vitro, that is in the microbial media as well as in the food products. We should evaluate the activity of a potential antimicrobial against multiple strain of pathogens, because in the many times in the food they contain multiple microflora. So, how this antimicrobial or bacteriocin etc., they are effective on multiple microflora. This information, this data should be generated, this should be evaluated, and this will make the use of antimicrobial more popular. Most antimicrobials are bacteriostatic rather than bactericidal. So, once this effect of that antimicrobial is reduced, the bacteria may again revive. So, applying the antimicrobial in a food involves either a model food system or the actual food, and this system demonstrate potential interference by food components, but allow for easier sampling by the researcher. The microorganisms utilized should be a natural contaminant or a pathogen of interest, and incubation conditions should reflect use and abuse. The success of application testing should be determined by increased shelf life or reduction of potential health hazards. So, we have discussed that is what are the different antimicrobials, permitted antimicrobial in different countries, how their effectiveness can be evaluated. There are certain antimicrobials or bacteriocins at present, they are being used in the some of the food products in some countries. However, there are still certain issues which should be resolved or which need more elaborate studies for making these natural antimicrobial more useful or to its application in food more common, and these are the activity spectra and application data, that is the microbial and particularly the mixed microflora, as I told you earlier, that is the data, and there are not much data available in the literature that is about the activity spectra of the mixed microflora, or how, that is when the antimicrobials are, how they get influenced. So, generating these type of data obviously help in using or making this process or antimicrobial use in food more and more popular. Okay. Then purification, that is of course, there is a the use of the purified this antimicrobial purified form, but many times it invites legal or regulatory problems, because these purified bioactives or antimicrobial might be considered by the regulatory agencies as a chemical, it might be compared as a chemical. So, more stringent data might be provided. So, maybe one form of its use may be that it can be that is like cultured butter, cultured whey milk products, etc., like that similar products where this in the known food materials these microorganisms can be grown and these cultured products can be used. So, of course, these aspects of that is the data also need to be generated. Then as I told you, combination of microorganisms. So, even effectiveness of combination of different antimicrobial when in a system, maybe that one microorganism, one antimicrobial may be more effective against one group of bacteria or microorganism, the other antimicrobial may be more effective with the other group of bacteria. So, this type of data also generating the data of the combined effect of different antimicrobials and that is reducing the microorganisms, etc., will be of great immense. Then resistance development, another important issue, that is many a times these microorganisms develop resistant to the chemicals, etc., after repeated or frequent use and after a long time, or even after their exposure to these chemicals for a little more time. So, this again it becomes a probability. So, some sort of that is research or that is the issue need to be studied and provide the data regarding their resistance and the microbial, etc., should be toxicological data and regulatory approval. I already discussed that is in the purification arm, and that is at the level at the dose level required for to be added into the food for providing the desired effect in the food. At that level it should not provide that is the toxic effect, because there are many naturally occurring, that is herbs, etc., or plant extracts, etc., they have the potential antimicrobial effect, but many of them some of them might be toxic otherwise. So, all these things, that is the plant extracts, etc., are systems which have antimicrobials, their toxicological data should be, and the cost, of course, it is very important factor, that is cost of using. So, like if this is to be used in a purified form, so it might incur more, it might become a costly process. So, it may result. So, of course, one way of that is using it involving less cost is to use it in the extract, maybe plant extracts of edible materials like spices, essential oils, etc. However, their concentration should be used in such a way that it should not influence the sensory effect of that, is because the plant extracts, spice extracts, oleogens, etc., they may result into the influence above a certain level, they may influence the sensory characteristics of the food. Even activity validation method, that is the activity of these antimicrobials or activity of the microorganism when they are in contact with the antimicrobial, etc., that is how it should be. There should be proper method should be developed for the validation of their activities. So, some of these issues in fact are there, the key issues which need to be resolved, and if the more and more data should be generated in this effect, and this will obviously influence or it will positively affect the use of these natural antimicrobials as a food preservatives or this biopreservation of food, which has a great potential in producing that is safe and good quality food materials or healthy food materials, because many times with these natural antimicrobials they also have many health benefits. So, the use of these natural antimicrobials or biopreservatives is a promising method or promising way in improving the shelf life as well as quality and health value of the food products. Thank you very much.